Hi, and welcome. So for those of you that are new here, my name is Ivan, and the focus of this channel is to more or less document my progress selectively breeding guppies. For those of you that have visited this channel before, you've obviously noticed that this format is a little bit different. And the reason for that is it's January, or hopefully by the time this uploads, it's still January. And I figured I might as well do a video where I'm reflecting on the things that I've done in 2024 and tell you about what my plans are for 2025. And I'm excited about what I have in store, and hopefully that can relay to you and you could be excited about that as well. I apologize for the lack of video consistency lately. I took a much needed break, but hopefully I'll get back on track. So before getting into 2024, 2025, I figured I'd give you guys a little bit of background, basically why I started YouTube in the first place. I'm going to go into a little fundamentals of genetics. I feel like that it might be a little bit necessary, but it won't be an in-depth video. It'll just be some technical terms that will be helpful when I start talking about some of the crossings and offspring that I've seen in my 2024 breedings. Then I'll get into my 2024 review through the lens of challenges. I'm not going to go into the nitty-gritty of each cross and the genetics of it. You could go to my channel and look at every individual cross for that. So I'm just going to look at some of the challenges that I faced and some of the solutions that I come up with, or at least how I overcame them. And then I'll finally get into my 2024 plans. So before really getting into it, I just want to explain that I do have a bit of notes on the side. There's a lot of things I want to get to and a lot of things I want to talk about. In order for me not to get too bogged down in the details, I just have a little bit of a guideline for me. This is different than my other videos, which are heavily scripted, so that way I could be as concise as I possibly could for those videos. This is going to be a little bit raw. I've tried recording this several times already, and it's complicated. It's a lot harder than I thought. So I just decided I'm just going to wing it. If there's mistakes here and there, so be it. We're just going to go through it, and hopefully you guys will stay with me through the end. Alrighty. So first thing I want to talk about is why YouTube. So let me write that down here real quick. The reason being is that I find a lot of value, or I found a lot of value in some of the documentation that I've found in the past where people have selectively bred guppies and recorded what their offspring look like. So what did their parents look like? What were they after? And then eventually what their offspring looked like. One of the channels that comes to mind that I took a lot of inspiration from is, and I'm going to pronounce this wrong, but Kaimuki Backyard. If you haven't checked it out, please go. It's really fun to watch him go through his backyard where he has tubs of guppies, and he would say, he would show you his males and females and then progress through his selection to basically start up a new generation, and so on and so forth. There are other websites that are not specifically related to guppies that I found very valuable as well. This is specifically beta territory. And this website honestly has some of the best documentation of spawns I have ever seen. And I enjoyed that a lot. And I wanted to emulate something similar. However, I wanted to do it in a video format. I believe video format conveys a lot more information, and there's a lot more detail involved in it as well. And so I wanted to do something, not just for you guys, but for me as well, because it's a journal, more or less. It's something that I could always reflect back and see what I did in the past and what the results look like. Before I officially started uploading, I did want to have a little bit of guidelines that I followed, objectives, more or less, or if you will, for what I wanted the channel to convey to the audience, or just for me for each video. And this is something that I'm consistently working with. I'm not perfect, but I think it's getting better with every video. So let me get through some of these objectives or guidelines. And I'm going to go ahead and list them out, and then I'm going to go one by one with each one and explain what I mean by each of them. First one is that I wanted the videos to be relatable. Second, focused. Third, credible. Fourth, digestible. Fifth, fun. Alrighty, so let's go one by one and discuss what I mean with each one. When I say relatable, I wanted at least the very beginning of this project to be something that a beginner hobbyist could find connection with. So at the very beginning, I wanted to make sure that I started with fish that were easy to find at any local pet store. Oftentimes when you think or hear people say, oh, if you're going to start breeding guppies, you want to start with high-end guppies so that way you could breed quality guppies. That is very, very true. But I think it alienates a lot of people that are just curious. What will happen if I take this fish and what will the offspring look like? Granted, I'm not going to be able to get through every single permutation of guppies, combinations, so on and so forth, but I wanted to talk about just some genetic principles. What can you potentially expect? You might not be able to expect a particular color, but at least you could expect whether there's going to be uniformity or if there's a dominant trait that you could recognize. And I wanted to do that with fish that were easy to get your hands on. And that's why partly, or actually the majority of why I started with snow whites. This is a fish that I felt like if you go to any store or big chain stores, you could easily find it. Granted, you can't really find the females. So I'll get to that in a little bit. Secondly, I wanted it to be focused. And specifically what I mean by that is, at least for the very beginning of this project, I wanted to really just focus on one goal at a time. And if you've watched my videos already, that one goal is just to restore that white color. I wanted it to be focused because oftentimes when I went and looked at projects that other people started to try to create a new strain, it becomes a little ambitious. It's a great goal and some people are really, really good at doing that, but it involves a lot of different characteristics. And when you do that, it gets really, really complicated. And I think a lot of people get bogged down with it a little bit, and the project eventually kind of fizzles out into a fancy mutt strain that doesn't really breed true. It may or may not, but I think oftentimes it does. So I just wanted to focus on one characteristic at a time because it makes some of the more complicated topics a little easier to understand. Thirdly, I wanted my videos to be credible. I'll be the first to admit that breeding and specifically selectively breeding guppies, I'm not an expert in this. There's a lot of genetics and a lot of new things that I'm learning along the way. So how do I establish credibility? So this goes to something that is often done in research. If you're starting up a new project in research, it is very, very helpful to start by reproducing an already existing experiment. So this does two things. One, it is something that is a lot more quantifiable. You have a tangible way to see whether or not you're doing something right because you have a way to measure how close your results are to what is already the standard or published or accepted. Secondly, if you're new to this project and you want to reproduce these experiments, it'll require you to learn the different techniques, all of the other equipment along the way. So how do you use it? How do you apply the theory behind it to get to the result that you want? So with all this in mind, I figured to do something in that nature, the best way for me to do that is not to create a new strain, but rather see if I could restore an already existing strain. Again, that'll be a very easy way for me to quantify how successful I am, because I could look at my offspring and see, well, how close do they look to the original strain? And then secondly, it'll require me to use different breeding techniques, and then it'll one, show you guys me using these techniques, and also teach me how to use them properly along the way. So that's what I mean by credibility. Also, just as a side note with this credibility aspect, I do want to mention there is a video that was uploaded by a guy named Alexander Williamson. He has a channel called Fishkeeping. Anyway, and he recently uploaded a video that is very similar to this topic of restoring a strain. It is from a different perspective, and so the video is called How to Create Any Type of Guppy When You Only Have Male Fish from Their Line. It isn't restoring a line, but it is putting in a lot of effort to make more of that one particular male, or maybe a line of males. So it's similar to what I've described in terms of restoring a line, but it's from a little different perspective. And if you haven't seen that video, I highly encourage that you do so. Alrighty. Next is digestible. I wanted my videos to be educational in a way that's not just a theory, right? I could upload several videos that talks about this is how you line breed, this is how you backcross, what is outcrossing, what is crossbreeding, and then that's it. There are plenty of videos that are out there that do that. I wanted these to be practical. I want to talk about it, but then alongside me talking about the theory, I'm actually applying them, and you could see tangible results. So this is what I mean by digestible. If I'm going to talk about something, I want to make sure that I also do it and show you guys how it's done. Lastly, I wanted these videos to be fun. And this is something that I'm definitely still working on, because it is very hard to take data and progress reports, so to speak, and documentation and package it in a way that is entertaining in a YouTube format. I think I'm getting better and better with every video. However, it is challenging. There are some complicated topics, and I do fumble sometimes, but hopefully they're packaged in a way that is a little bit easier to digest, but also fun. Alrighty, so with all these things in mind, I set out to find the fish that I would start this project with. So I wanted it to be relatable, so it's something that I would find at a local store very easily. And obviously I landed on snow white guppies. And it also helped that I was prone to just solid colors. And so it was an easy choice for me. Focused in the sense that I'm going to restore this white color using random fish. That was also not that hard to do, because whenever you go to a local pet store, specifically with snow whites, it's actually kind of hard to find snow white females. So the project kind of landed on my lap. Well, if I can't find these females, I might as well start jumbling these genetics together to see if I then eventually can restore this snow white phenotype. And then eventually I would probably have a pool of females that are snow whites as well. That was the initiation of the idea. And so at the beginning of, or maybe in the middle, sometime in February 2023, I bought myself 8 different guppies. These 8 guppies were 2 snow white males and 6 random females. After a cycle of meds, there were a few that died. I think 1 male died and then 2 females died, which left me with 1 snow white male, who we eventually named Gandalf, and then 4 random females. And this was the very beginning of the project. The very first challenge that I encountered is the very pesky adaptation of female livebearers. They can store sperm, which means that once they've mated the first time, they could produce fry every single month like clockwork. And this is problematic when you're trying to selectively breed guppies and you're introducing a brand new male. You have no guarantee that the offspring are all fathered by that male. And so it makes the statistics actually really hard to deal with. So that was my first challenge, because I did buy these fish from a local store where they didn't separate their males and females. Granted, the snow whites were in a different tank, but the females, and there was no telling whether they were virgin or not. However, I tried to purchase very, very young fish. So the way that I thought I was going to tackle this problem was, well, if they're already pregnant, let me just let them drop their first brood. I will raise this brood and separate the males and females, and then start my project from those juvenile females. However, my first batch of fry didn't show up until May 2023, so a full 3 months after I've already bought these fish. And I was already antsy to get this project started, so I just assumed that these females were virgin, or at least their offspring was fathered by Gandalf 100%. And I figured that, hey, if my assumption is wrong, I'll be able to tell based on the genetics of the offspring. And then that'll dictate whether I was justified in making that assumption or not. Long story short, I think I was justified. And I might get into it a little bit, but the phenotypes of the offspring, both in the first initial crosses and the back crosses, suggested that all of the fry, or all of the offspring from these initial crosses, were indeed fathered by Gandalf. Okay, so in this part of the video, I want to get into a little bit of genetic housekeeping. I think it's a little bit valuable for some of the discussions that we're going to have later on in the video. However, this is not going to be an extensive discussion on Mendelian genetics. There are plenty of videos on YouTube that do a much better job than I can on really teaching you this stuff. One of the reasons I do want to get into it a little bit here is because I may know the fundamentals, but I didn't know the specific genetics that were involved with the white phenotype of a guppy. So I want to just kind of hash it out a little bit, and then before we get into my guppies and their white phenotype. So here I'm going to spend some time on an example that is completely made up. I have a guppy that is gray. So this isn't new. We have gray guppies, and so this is what I'm going to call my wild type gray. Let's say in my made up scenario, there also exists a guppy that is cyan color. And let's say these are the only two different color types that exist that goes throughout the entirety of the guppy body. So every guppy, and most living things for that matter, inherits DNA from both parents. And the DNA is just a set of instructions that are all wrapped up in these things called chromosomes. So you get a set of chromosomes from the mother, and you get a set of chromosomes from the father. And they kind of separate into two categories. You have your sex chromosomes, and then you have your non-sex chromosomes, just called autosomes. This means that, at least for the non-sex chromosomes, you have duplicate instructions for every gene that you could get. But they are not necessarily identical instructions. So these variations in instructions, or these variations in these genes, are what we call alleles. Naturally, the question comes up with, what if a guppy inherits two different versions for the same genes? So just two different alleles. In the simplest case, one allele just overrides the other. So this means that one of these alleles is dominant compared to the recessive allele. Again, I want to stress that this is the simplest case. There are different influences, different ways that these alleles could potentially interact with each other, leading to codominance, incompletely dominant, but I don't have the time to kind of get into it, and it's sufficient for right now to just talk about the simplest, just dominant and recessive case. So what does this have to do with my example here with the wild type gray and the cyan guppy? So let's say that the color of the guppy is controlled by a single gene. And we obviously have two versions. So this would be two different alleles. One allele that controls the color to make it cyan, and one wild type gray. Alrighty, let's say that the cyan color is the recessive allele. And I will note this with a lowercase letter c. Let's say that this allele is dominant, and I'm going to note this with a capital letter C. And I know C's get a little bit weird. I'm going to underline it. Okay, so like I talked about earlier, each guppy inherits DNA from both parents. That means each guppy is going to have two alleles for the same gene. And if one is dominant, it's going to override the other. So if cyan is recessive, the only way that it could appear cyan to us is if the other allele is also recessive. And to make it simple, at least for now, we're going to say that this guppy here also has the dominant allele. So they both have matching alleles. So because these guppies have matching alleles, this is what we call homozygous, meaning the same alleles, homo same. If a guppy happens to have a mismatch of alleles, a guppy inherits a dominant allele and then a recessive allele. This is what we will call, or is called, a heterozygous guppy. And this guppy, because this dominant allele overrides the recessive one, the wild type gray is what we will eventually see. So this brings up the difference between when I say genotype versus phenotype. So genotype is the genetic makeup, the combination of alleles that a guppy has inside their chromosomes, so to speak. So in our case, we have 2 uppercase letter C's, 2 lowercase letter C's, and then a combination where we have a mismatch. So this is what we would call a genotype. And in my example here, I have 3 distinctive genotypes. Now, a phenotype is how this genotype is manifested or expressed physically in a way that we can see it. In this example, the way that it manifests is the overall color. And so we only have 2 colors. We have the cyan and then we have the wild type gray. Even though we have 3 different genotypes here, we only have 2 distinctive phenotypes. This becomes relevant later on because this heterozygous expression gets a little bit complicated when you're trying to select guppies, because it kind of throws out offspring that, if you didn't know they were heterozygous, can get a little confusing. Okay, so this channel is about selectively breeding guppies. So how can I use this in my experiments? How can I use an understanding of genotype and phenotype to predict the offspring of any parent that I use? Obviously, it's not going to be 100% accurate, but there are tools that can help us get really close to predicting what those outcomes are. And one of those tools is called a Punnett square. I'm just going to illustrate a simple case where we're using these 2 genes of just the overall body color, where the phenotype is either cyan or wild type, gray. And that means that the Punnett square is just going to be a 2x2 square. So let me just go ahead and draw that out. And then we look at the parents. Usually the way that I do this is I draw out the male up on top and the female on the side. For a single gene, there are 2 alleles that each guppy has, and they could pass down one or the other, but not both at the same time. So for instance, whatever gene that we're looking at, say it's just XX, the male will pass down either one or the second X. Same with the female. I know I wrote out X's, but let me now go back to the example that we used. And I'm going to start off with the simplest case and then work our way into higher complexity. So we're going to swap out this genotype. So let's start with the simplest case where both of our parents are cyan. So the way that I do it is I start with the genotype, and I split the genotype into its respective columns and rows. So for the male, this would be the 2 up here, and the female would be the side 2 here. Then, in order to fill out this Punnett square, you then bring the allele down from one and then across from the other. And you do this for every single square. So again, this very simple case where both of the parents are cyan, this Punnett square is telling me that all of their offspring we can expect to also be cyan. And I could do this with 2 wild type gray parents that are homozygous. So really quick, what that would look like is these 2 dominant alleles, we split them up into their respective positions, and then we start filling out the squares. Again, the expectation here now tells us that we could expect that 100% of the offspring will also be this wild type gray color. Now, it gets really interesting when we start combining the different phenotypes and genotypes. So let's go through all these one by one. So I went through the first 2 cases, and there are another 4 cases that I want to go through. So let's do that really quick. So the next scenario is something that I think everyone would want to try first, and that's a pairing between opposite phenotypes. So let's say we have one parent that is homozygous, gray, and the other one is going to be homozygous, cyan. So again, we're going to drop these to their respective positions and then start filling in the Punnett square. So I'm going to start with just the black marker first and then filling in the cyan. All right, so what does this Punnett square tell us? Well, it tells us that the offspring all genotypically are very different than the parents. However, they are all the same amongst each other. What does this say phenotypically, though? Well, this dominant allele overrides the recessive one. And we know that in this case, these offspring, 100% of them is going to look like the wild-type gray, but they're going to be heterozygous for it. All right, this is going to become important, so keep this in mind. I do have a figure that I'm going to show later on, but just keep this scenario in mind. The next example is if we take a heterozygous offspring or a fish and pair it to a fish that is homozygous either cyan or homozygous gray. In this case, we still have a cyan guppy, but we're pairing it with a guppy that is heterozygous gray. And this is where it kind of gets complicated. Sometimes you don't know that this fish is heterozygous. So what does this look like? Again, we split these to their respective positions. We have cyan, and then we start filling in the squares. So I'm just going to go down, then down, then across here, here, and then our top row here is where we have the dominant allele. So what this says is that even though we have parents that have two different phenotypes, they're also going to produce offspring with two different phenotypes. So you have, in this case, 50% of the offspring that will have a homozygous expression of cyan, and then 50% that will phenotypically look like they're gray, but their genotype is heterozygous. The other case scenario is where you match a heterozygous gray with a homozygous gray. I'm not going to go through it. Basically, it's a split between genotypes, but the phenotype all looks the same. I want to go ahead and get to case number 6, is where we have two fish that are heterozygous. This is where things get a little interesting, or at least more interesting than what we've seen it as so far. Right? So let's say both fish are heterozygous, and we'll fill in the respective positions for the alleles, and then start filling in the entirety of the Punnett square. So we bring this down, this across, this down, and then start filling in the recessive alleles, right? Okay, this is probably the most complicated yet also the most interesting scenario. We have two fish that phenotypically are gray, but genotypically are heterozygous and carry both alleles. And when we look at their offspring, this again shows us the distribution and the likelihood of this occurring. We have 3 different genotypes. We have genotype number 1, where we have a guppy that inherited 2 dominant alleles. So genotypically they are homozygous, and phenotypically they are wild-type gray. Our next genotype is a heterozygous expression of both alleles. So genotypically, there's a mismatch in alleles, but phenotypically, they also look like the wild-type gray. And then our last combination here, I'm going to go in blue, is the cyan guppy. So genotypically, they're homozygous, and phenotypically, they're cyan. So in this distribution, if we look at genotypes specifically, we could expect our offspring to have, we expect 25% of our offspring to be cyan. We expect, 2 boxes out of 4, 50% of our offspring to be heterozygous but wild-type gray, and then 25% of our offspring to be homozygous gray. Again, this is genotypically, but what does this mean phenotypically? Well, we know that phenotypically the homozygous gray and then the heterozygous genotype both look the same. So in this scenario, we have a whopping distribution of 75% of our offspring to phenotypically look like the wild type gray, and only a small subset of our offspring to carry and show the cyan phenotype. Whew. So I know this was a lot, but I feel like it was important to kind of go over this because they are relevant for some of the discussions I want to have with my crosses so far. And I'm not going to draw these out again. I went ahead and printed out the relevant cases, and so I will reference these again when the time comes. But this still begs the question, okay, I understand the fundamentals, right? I use this cyan example, but what about the actual genetics of the fish that I bought, like the snow white guppy? Like I didn't know what the genes were involved, and it was actually kind of hard to find information for it. It wasn't until Nick from Keeping Fish Simple uploaded a video where he toured a fish room from Glenn. And Glenn kept a wide variety of different shrimp and guppies. And he passed a snow white tank and just mentioned that, hey, the white color is actually a result of magenta and Storsbach. This is exactly what I needed, because now I could utilize the resources that I had available, specifically Alan Bias and Philip Shattuck, as well as Stan DeYoung, and I could really hone in my research. And I could look at magenta, I could look at Storsbach, and then maybe other genes that are involved, and then actively seeing them happen in my own breedings. So, Glenn, if you are watching this, please start uploading some long form videos. I think they would be bangers. Your enthusiasm is infectious. I loved seeing the tour that Nick did with you, and I'm sure a lot of other people would as well. I guarantee you will find an audience. Okay, so with that out of the way, let's get into my 2024 review and look at the past year through a lens of different challenges that I've faced. So I uploaded my very first video in the middle of December of 2023. Yes, it's not 2024, but close enough. Throughout that year, I've introduced and set up 15 different crosses. So let me go ahead and write that out. All these 15 could be categorized, I think, in 3 different groups. So this first set is the initial backcrosses. This is the very first pairings that I did with Gandalf and the purchased females. The next set would be the backcrosses, and this spans crosses 5 through 10. The last group are crosses 11 through 15. And this is what I'm calling intercrosses. So these are crosses that no longer involved Gandalf. This is now spanning crosses of the offspring within the backcross generation. Okay, so let's talk about the first set of groups, so the initial crosses. Obviously, the whole project stemmed from trying to restore the Snow White phenotype, but I needed to start with random females. And so this is just the simple, okay, I have this fish, let's cross them and then get some offspring. So the challenges that I faced with this was that I didn't know the genetics of either parent. Again, like I alluded to, I didn't necessarily know all the genes involved for the Snow White phenotype, but let alone know anything about the females. They were completely unknown to me. The only thing that I figured I could go by is that they perhaps were homozygous for whatever genes that they have. Maybe they came from a pure strain initially, and these genetics differed from Gandalf. So yes, they're homozygous, but they're homozygous in a different way. So this is kind of what I alluded to, is this chart here when I did the Punnett square of a homozygous cyan and a homozygous wild type gray. And so with that assumption in this prediction, what I thought would happen is that when I crossed these fish together, then I would expect that their offspring all looked at least uniformly the same. There's a lot more genes involved than just the one, but I felt safe to do so because, yes, there were a myriad of different genes involved, but they were homozygous. And when they recombined, yes, their recombination is very varied, but they're all varied in exactly the same way. And it turned out that this actually was the case, at least for crosses 2, 3, and 4. They all had more or less uniform color and pattern amongst their siblings. The only exception was cross number 1. So if you remember, or if you've seen these initial videos, I had 2 distinctive phenotypes in that cross. So naturally, the first thought is, okay, fine, that female wasn't actually virgin. And that subset of offspring were fathered by some unknown male that wasn't Gandalf. However, after looking at some backcrosses and looking at those offspring, I don't think that is actually true. And I'll get into that in a minute. So the other explanation potentially could be is that there's some other recessive gene that is at play. I think the split was something 60-30, with 30 being this oddity. And that kind of falls into the category of maybe some recessive gene. However, I haven't figured that out yet. But I do think there are some consequences of that later on. And I won't get into that in this video. This will be in the next update video, I think on cross 13. The next group of crosses is this backcross set. This is where things got a little bit different and interesting. And that is because, again, if I bring out my cyan example, in these initial crosses, they're all heterozygous, and they're all heterozygous in the exact same way. Now, if I take this offspring and breed it back, like I did to Gandalf, say in this example with cyan, now I get a mix of different phenotypes that I could expect. Obviously, in this case, in this one single gene, it's a 50-50 split. But if we then extrapolate to a bunch of different genes that were involved, we then get a myriad of different phenotypes. And in my most complicated crosses, I think this was cross 6 and cross 9, there were something like 16 different phenotypes. And it's a function of heterozygosity and then the number of genes. I think in those cases, I had 4 genes, and so that's like 2 to the power of 4, but we're not going to get into that here. The main point here is that there are a lot more phenotypes than my initial crosses, but they could be a little bit predictable. So in these back crosses, this is when I started getting a little more familiar with the genes that were involved. And so I'll go ahead and list them here. So the first one, it was base body color. So I'm just going to say BB color. And this has a very clear visual phenotype. You have either a gray guppy or a blonde guppy. And the blonde phenotype is the recessive. Second is magenta. Magenta is a dominant gene, or incompletely dominant depending on what resources you use. But for the most part, across my initial and my backcross categories, I treated it just like an autosomal dominant gene. Third was Storsbach. And this is a recessive gene. So Storsbach gave me probably the most problems. And that's because this was something that was really, really hard for me to identify in my females. However, I think the solution that I came up with this was something that I stumbled on recently. When I was trying to film some really good B-roll for my videos, I got my hands on a much brighter light, so that way the camera work was a little easier, so I didn't have to bump up the ISO so much. And while I was looking at the camera through the viewfinder, I noticed that because of this higher intensity light, the reflection of this iridescence was a lot more prominent. So I think I have a way to solve this issue moving forward in my next few crosses. The next gene is European Blau. And this is a recessive trait. And so for the sake of consistency, let me just get that out. Okay, so this is something that I'm still not 100% sure. There's, I think, several different Blau genes. I think European Blau is what's involved here. Again, this gene, based on Alan Bias's chart, kind of removes different red pigmentation at different levels compared to whether it's on the body or on the fins. But there are a lot of other genes that do similar things too. I think it is European Blau, but it may not be the case. However, it's I think based on some of my crosses so far, I'm going to go ahead and assume that that is the case because it's been helpful so far. Lastly, and this is not really involved with snow white color, but this is a gene that was carried from my initial females, and that's the half black. But this is a gene that is controlled by the X chromosome, or at least the version that I am assuming my fish have, and it is dominant. So it's NI, first version. Okay. So obviously, this is a little bit more complicated than the autosomal dominant scenario that I talked about. However, because it has been only carried through on the X chromosome and I only used Gandalf, it behaved more or less like an autosomal dominant gene. I want to probe into this a little bit more. It is veering off what my initial goals are, but I'm not going to spend too much time. Although I am setting up a cross where the male is now the half black carrier and the female isn't. So it'd be really interesting to see if this assumption here is actually what's happening. Okay, so apart from the actual genes and understanding their inheritance patterns, the other difficulties that I've encountered with these back crosses was cross 9. And if you've seen my videos, you know what I'm talking about. So cross 9 had an issue where my females wouldn't produce viable eggs or viable fry for months. It took, I think, several months before they even produced swimming fry, and that was encouraging. I thought, great, I got fry, the project can move forward. However, as the fry matured, almost all of them turned out male. There are things that are rattling around in my brain that it could potentially be, one being that it's a lethal gene involved here or there, maybe some temperature, but I'm not convinced one way or the other because there's just not sufficient amount of data for me to be convinced. During this whole saga, it questioned whether or not Gandalf was viable. Obviously, I got fry from cross 9, but I set up an additional cross 10 to test whether or not Gandalf was viable. Turns out he isn't anymore. Cross 10 was set up way back in April, and I have never gotten any viable fry. And actually, only recently did this female pass away. So cross 10 is no longer something that is going to be contributing to the overall progress. The intention of the back crosses were to get as close to the white phenotype as I could. And every single cross from 5 to 9 produced offspring, or at least a small subset of offspring that were indeed white. That confirmed that my initial assumption that my 4 original females were indeed bred by Gandalf was true. Again, this has to do with this scenario here where I'm combining a heterozygous offspring with Gandalf, who is homozygous. There's an argument that says maybe not, but I think the evidence suggests otherwise. I am seeing white guppies in all of these back crosses. So let's move on to the intercrosses. Gandalf was no longer viable. That means I can't use him anymore. So in these intercrosses, this is where I started taking offspring from my individual back crosses and intertwining them together. So let's go through them a little bit one at a time. All right, cross 11 was a pairing between cross 5 and cross 6. So I'm going to go ahead and mark those as done here as I go through with them. This cross has produced a lot of offspring. And honestly, I think right now this cross is the closest one to producing 100% snow white guppies. So this is a good thing. The next cross, cross 12, was a cross between 5 and 7. However, this cross actually never produced any fry. I'm not sure exactly why, but I didn't want to pursue it too much because I'm using cross 5 twice here. So I just chalked it up to females that were not viable. But in order to salvage cross 7 and not lose it, I established cross 13. So cross 13 was a pairing between a gray-based Snow White male from cross 7 with 2 females from cross 8. So let's go ahead and cross those off as well. Now, cross 13 is just a package of problems. Maybe not so much big problems, but it presented a lot more challenges than what I anticipated. So let me just go over what those are. The first thing that happened was that the male died. It is very unfortunate. It was an oversight on my part, but I'm always exploring different ways to provide cover for my fry. I don't want to say that I have the best solution to that problem, but I'm constantly trying to evolve in how I'm doing it. And so in that particular cross, I had this basket here that I would invert to allow fry to swim through, but the parents would not be able to. However, the holes in this basket were just big enough to have the male's head pass right through, and he was not able to get out. And so that ended up killing him, which is unfortunate. So that's the first problem. However, I was lucky enough that the females bred with the male, and so I was able to get offspring pretty regularly from that cross, and so it wasn't completely lost. The next challenge with cross 13 involved the magenta gene. Up until now, I kind of ignored it because it was a dominant gene, and so all of my guppies up until now were carriers, so it really didn't matter. However, when I'm starting to mix these lines, because I didn't know whether a guppy was heterozygous or homozygous for it, there's a chance that it could reappear in a guppy not actually carry the magenta gene. For instance, in this example with cyan, where I have 2 guppies that express the dominant allele, but when I mix them together, all of a sudden I get the phenotype of a guppy that doesn't even carry that gene, in this case, this dominant allele. And so that could potentially happen in this cross. So that's a challenge that I foresee, but I don't have a clear solution to that yet. I'm still working that out. Storsbach is the next one. Again, like I mentioned earlier, it was hard for me to identify females that properly express this trait. I have found a solution for that, but this cross was established before then. So just like with magenta, where I didn't know whether my parents were heterozygous, there's a case that it could be happening with Storsbach as well. Lastly is I used 2 females. Usually, at least leading up until this point, that wasn't that big of an issue. Again, the pool of fish that I was pulling from, yes, they were heterozygous, but they were heterozygous in the same way. That's no longer the case in these intercrosses. There's a potential that the 2 females that I'm pulling, they might phenotypically look similar, but they genotypically might be different. And therefore, they could throw offspring that statistically are different distributions amongst the broods. So that's cross 13. So up until now, this is probably my most complicated cross. It involves a lot of unknowns that hopefully I could tease out later, but keep an eye out for that update video. It's going to be complex. It's going to be a lot of data, but hopefully I could package it in a way that is digestible and fun. I haven't finished describing all of my crosses yet, right? And the reason being is that I think these intercrosses can be split into 2 additional groups. So that would be between crosses 11 through 13, which are specifically designed to chase after and solidify my first initial goal of a line that breeds true for the Snow White phenotype. But these next 2 crosses are a little bit different. I am chasing an iridescent forehead trait. So this is something new that popped up. And I like this trait so much that I decided to try to set aside some additional crosses where I'm chasing this trait. So here I have 3 different guppies, and here is the male of interest. It's a little hard to see in this photo, but I might put a little overlay if it's easier. But his forehead has a lot more iridescence compared to this gray-based Snow White male and this blonde-based Snow White male. And I want to try to infuse this trait into my overall Snow White line. So these additional crosses are focused specifically on that. So cross 14 is between two siblings. So this is a male from cross 8 crossed to a sister of cross 8. So this male is the one that holds the iridescent forehead trait, and we named him Thorn. And I figured I have the highest chance of producing more males that have the iridescent forehead trait if I paired him with a sister. Now, cross 15 is between Thorn. This is a cross 8 male. And now I'm diving into my intercrosses, cross 11. So like I mentioned earlier, cross 11 had the most guppies with the Snow White phenotype. So I figured I might as well start infusing the iridescent forehead trait into the Snow White characteristic in general. Even though these guppies may not express it, I at least wanted to have a pool of guppies that were carriers. I didn't actually officially introduce cross 15 yet, so keep a lookout. It'll be a fun one. Okay, so that basically wraps up 2024. There was a lot of crosses, there was a lot of successes, and a lot of confusion at times, but I think it's bringing us a lot closer to my final goal of establishing a line that finally breeds true for that Snow White phenotype. So now we're diving into 2025 and what my next plans are. So I'm going to start listing them out here. My primary goal is still trying to fix a line that breeds true for the Snow White. But the question now is what constitutes a success? Is it if I have one cross that produces 100% Snow Whites, is that considered job well done? Did I achieve my goal? Maybe, but I think there's a lot of value in having replicates. So if I could do a cross that produces 100% Snow Whites 3 times, I think we could safely say that I've achieved my goal. The second goal would be to incorporate cross 9. So I've already erased it, but I'm going to backtrack on my film and show that cross 9 hasn't been paired off yet. And this might be a mistake because it was a little problematic in terms of viability and skewed more towards males, but I think there's a lot of value in the genetic diversity that cross 9 can bring, specifically that Snow White male with the half-black trait. He has a very beautiful white dorsal fin with a lot of volume that I think would be good to incorporate into the overall pedigree. The next thing I want to do for 2025 is consolidating my pedigree. I have a limited amount of tank space. It's not infinite, so I really have to be mindful of the number of crosses that I establish. The way that I want to do this is, I think, a little bit different. Maybe it's been done before, but I haven't really seen it described elsewhere. I don't want to maintain my lines through line breeding or any of the other conventional methods. I just want to consolidate my pedigree to maybe 3 sort of lines. Okay, let's say we have 3 different pairs, and I'm going to represent them with these boxes here. I then would like to take a male from here, a female from here, and so on and so forth, basically pairing each adjacent male and female from these sort of lines. And when I get to this last one, it just carries over here. And then I want to do this over and over and over again. And essentially, this looks very braided. So that's why I started calling it a braided breeding technique. I'm not sure if anyone's done it before, but I feel like this method decreases the amount of inbreeding that could happen compared to, say, line breeding. And this also gives it a good way to start infusing different characteristics in as well. And this also gives me a good pool of Snow Whites that I could technically use as backcrosses without backcrossing to the same male. It's just backcrossing to the same strain. The next one, and I talked about this earlier already, is the iridescent forehead trait. And like I've mentioned earlier with my intercrosses, this is something that I think is really, really neat. And if I could incorporate this into my snow white lines, I think that would look really, really cool. And lastly, and I think this is going to be the most exciting, is I want to start incorporating new characteristics, and specifically the half moon. I've already bought the fish that I want to do this with, and they are called half black red roses. I'm not too keen on the half black aspect of it. Again, this is a little bit small. Here we go. Yeah. So I'm not too keen on the half black aspect of it, but I do really like this half moon characteristic, as well as this high dorsal fin. And the reason I went with the red roses is because of the solid color of the fish. I find half moons a lot. They're not that uncommon, although they are a little harder to get than your typical guppy. But most of the time, these half moons exist on your red dragons or your blue dragons, or fish that have a lot of pattern. And I kind of want to avoid too much pattern because I feel like that would be a little bit difficult to then remove from the all white snow white phenotype that I'm working with. And I actually had a friend on Facebook tag me in a post where someone was talking about a magenta half moon. Here, again, let me give you guys a little bit of close up. So it tells me that it is at least somewhat possible. So the challenge here is whether or not some of the genes that already are required for snow white phenotype, whether or not they interfere with the half moon characteristics. Let me explain. One of the genes that looks like it's very necessary for a snow white phenotype is magenta. And magenta not only controls color and proliferation of different pigments, but also fin size. So if you've noticed, a lot of snow white guppies have a lot shorter finnage, and that's because of magenta. Now, the issue is because I want half moon and those are larger tails, the question is, how do these two genes then interact? Will I completely lose a half moon characteristic because magenta then shortens it such that I can't tell anymore? So this is going to be really, really interesting. I really like this half moon. This photo here suggests that it's possible, maybe, hopefully without all of this black speckling, but how, I'm not sure. Hopefully the reduction of fins doesn't stop it from spreading. I think the half moon, or at least the red rose, increases the total number of rays. So fin reduction doesn't equate to ray reduction. So hopefully that's still something that could happen in my snow whites, and I'm excited about. It's not something that I will be able to fully complete and incorporate in 2025, but it's something that I'm excited to start. And I have the guppies already. I bought 2 pairs. They given me offspring, and I've been waiting to see if they bred true. Again, I wanted to make sure that they were homozygous for the traits that I'm after. So that way it's easier to then predict the offspring when I start crossing them into my snow white line. I'll likely be crossing both a male and a female into my snow white lines because I think that half moon characteristic and likely that high dorsal is carried on the X chromosome. So it'd be really vital to do both the male and female and male crosses into the snow white. Okay, and I think that's a wrap. I want to again establish a snow white line that breeds true, that also incorporates my cross 9 that is still hanging and not crossed in yet. I also want to consolidate my pedigree so that way I have less pairs that I'm working with in this braided breeding fashion. I also want to incorporate the iridescent forehead trait, or at least understand how it's inherited. There's a chance that it may not work with the genes that are involved with snow whites. And then lastly, I want to start introducing new traits that I really like, particularly the half moon. The high dorsal is going to be a huge bonus if that occurs, but I'm excited about that. So this is a little bit of a longer video. But thanks for sticking through it with me. I think there was a lot of value that was discussed here. And if this is something that you find interesting and you want to follow along, please consider subscribing. There's a lot more that is going to be coming. I am hopefully going to start uploading regularly again. And one of the intercrosses, I forget exactly which one, but I think it is Cross 14, should be the next video, or the focus of the next video. And then I'll start subsequently going through each of these crosses, which will eventually lead up to the initial crosses that I will be doing with the red rose strain that I purchased. Thanks for watching, and I hope to see you in the next one.